So good afternoon, everyone. It's a great pleasure to be here. Thanks for this kind and generous introduction. So in this presentation, I'll give you an overview of some of our work on the use of computation for designing, for creating new kinds of microscopes, new kinds of sensors, and diagnostic tools. In terms of application space, we're heavily focusing on mobile health, on telemedicine and global health. And if you think about these application areas, they all share some common elements. Before I start talking about the science behind this kind of work, I'd like to go over mobile phones and the capabilities that make mobile phones so special, especially for computational imaging and sensing. And I'd like to start my presentation with this slide, which is comparing the Moore's law, the transistor count in our CPUs, against the megapixel count of our mobile phones over the last almost 2 decades. What you see here is an exponential growth. The megapixel count of our mobile phones has been doubling every 2 years, and it's now reached to more than 40 megapixels. This is a trend that is not expected, but it's so exciting for us because the instruments that we're creating based on mobile phones, based on CMOS imagers taken from mobile phones, will now enjoy this rapid growth and improve their performance in one way or another. I'll share with you some of our recent results that utilize these smartphones with more than 40 megapixels to even, for example, see single viruses or even single DNA molecules that are stretched and do scientific measurements with interfaces integrated with these smartphones. So that's quite exciting, and if you bear with me, I'll go over some of these results. In addition to the camera interface, the CMOS chip, there's another element of the mobile phone that's so special, and that is mobile phones can also process these images, these sensory input, rapidly because they have graphics processing units. It's fair to say that the smartphone is now a better supercomputer than early generation IBM supercomputers. That is so important because it's not just the camera interface, but also the digital processor that is quite advanced. That is all available to us because of the economies of scale that come with mobile phones. We have around 7 billion cell phone subscribers, and that's the reason why this infrastructure literally works everywhere, even in remote parts of Africa. And that's an infrastructure that we're relying on to bring new kinds of measurement tools. And in this slide, I'm sharing with you some of these examples, all of which are essentially 3D printed interfaces, optomechanical interfaces that's attached to a mobile phone or a smartphone in many of these cases, to convert the mobile phone into an imager, into a microscope, into a diagnostic tool. I won't have time to go over all of these, but it's now routine to convert your cell phone into a microscope, into an advanced microscope to the level where, again, you can even see sub-50 nanometer particles that are fluorescent labeled. You can look at viruses and even DNA that is stretched using disposable chips. And you can also use server connectivity to do machine learning and other types of analysis on these captured images. Microscopy is so powerful, and I'll share with you later on some other types of microscopes that leverage not necessarily the cell phone itself, but cell phone components like CMOS imagers to create these standalone devices to do high-end microscopy, gigapixel microscopy using field-portable interfaces as shown here. In addition to microscopy, there are other types of instruments that you can build around a mobile phone. This is actually an earlier generation of a smartphone, which is using an interface like as shown here to do imaging flow cytometry. This is another one that is an Android phone, has an application and an interface at the back for looking at fluorescent assays to quantify, in this case, urinary albumin concentration with a few parts per million level of sensitivity. Next slide, I show some other examples where it's the same theme to bring functions that you normally have in laboratories to remote locations to field portable implementations in a cost-effective and lightweight scalable interface. This is, for example, a blood analyzer, which is using fluorescence microscopy and bright field microscopy on the phone to capture images of blood cells, red blood cells and white blood cells, and using the interface Android application on the phone to quickly process these images to give you blood count under 30 minutes, including labeling steps and everything. Some other examples are shown here. This is an E. coli sensor. Toward the end of my presentation, I'll show you other types of waterborne parasite detection systems that also utilize server backend processing for looking, for example, Giardia, crypto, and other types of waterborne parasites. This is another recent work where we used here nanoparticles and color changes induced by heavy metals, specifically mercury, to create an interface that can, with a few parts per billion, nanograms per mil, quantify the presence of, in this case, mercury ions in drinking water using a very cost-effective interface, which is a few cents per test. And some other examples are shown. In addition to mobile phones, there are other emerging interfaces like smartwatches or wearables, in this case, like Google Glass. We've written one of the first biomedical applications for Google Glass to quantify lateral flow tests. These are immunochromatographic diagnostic tests that are imaged with Google Glass camera in a voice-activated and user in an easy-to-use interface. And all the processing and machine learning is done off the server to quantify the test of interest. It's not yes or no, it's actually a quantified reader that comes with the Glass interface. And recently we've used another application, found another application for Glass, which is in this case quantification of chlorophyll content in leaves without any damage to the leaf. It's also working in a similar way. It's based on imaging and machine learning. These are exciting because there are some examples how you can utilize the mobile phone, its hardware and its connectivity, its computational power, to do some of these measurements, excuse me, some of these measurements that normally are done in laboratories. I'll give you examples of that I haven't highlighted maybe toward the end of my presentation. And they share a common theme, cost effectiveness and field portability in an extremely compact and lightweight interface. But that's not the only thing, because these devices and these measurements are also connected to each other. And every time that they measure, they also place a stamp as a function of space and time. And that's another angle, because you're going to have a phenomenally large amount of data coming from various different places that these kinds of interfaces are being used to essentially look at trends as a function of space and time. It would be extremely useful, especially for epidemiology studies and looking at how things change and what are the cause-effect relationships. At this point, I'd like to also make a conflict of interest, a disclaimer. I'm also a co-founder of a startup company commercializing many of these technologies, licensed by a company that we started up in LA. It's Holomic. You can learn more about Holomic and its products. It's actually one of its first-generation products. It's a universal diagnostic test reader. It quantifies fluorescent and colorimetric diagnostic tests, and it also has the backhand server for processing and for tabulation of the results and integration with electronic health records. So we're also very proud because last week the World Economic Forum selected Holomic as one of the technology pioneers in 2015, which is among the most promising 49 early-stage technology companies in the world. So in that regard, some of this work is already in product form and being currently used in different ways, and also CE mark approved and FDA approval in the process. So I want to come back now to some of these interfaces and share with you the details and how you can make use of some of these designs, some of these instruments for various different measurement or imaging or sensing tasks. And I'll start with microscopy. It's so general, it applies to many things, and that's why I think it would be applicable, or it would be tangential to many of your research projects back in your institutions. So the microscopes that you see here, either attached to the mobile phone as shown here, or using standalone devices that literally take off the CMOS imager from the mobile phone and create a field portable interface as shown here. These are not traditional light microscopes. They're not using lenses or objectives or other bulky optical components that you normally find in a microscope design. In fact, they're lens-free, as a result of which they're extremely compact. And one key signature of lens-free imaging in the way that is shown here, these are high-throughput microscopes that can look at large areas and large sample volumes in a highly sensitive manner. So because of the lack of optical components or an imaging system, these kinds of microscopes that utilize the CMOS or CCD taken from a digital camera, they literally are starting with sampling of shadows. If you place, for example, take a CMOS off a mobile phone, remove the lens, you have this silicon chip which has, let's say, 5 to 10 megapixels. If you place a sample right on it, where the distance between the sample to sensor is, let's say, sub-millimeter typically, you'll be capturing these diffraction patterns, these shadows of the specimen, and it could work like a cytometer based on the signature. So if you look at, for example, whole blood, you'll be mostly seeing these shadows of red blood cells, and every now and then you would see a white blood cell. It could work like a cytometer, a blood analyzer, based on essentially computer vision and based on these shadows or library of shadows. But it's more powerful than that because it can also do reconstruction, because these are not dummy shadows. They're actually inline holograms created by scattering of light from each one of these objects and its interference with a background. So basically, you start with these dummy-looking shadows, and using Fourier optics and phase retrieval algorithms, you retrieve the phase that is missing at the CMOS and reconstruct an image, as shown here, except there is no optics, there is no literally objective lens or any other imaging system between the cells, in this case blood cells, and the CMOS imager, nicely matching to a traditional light microscope. This is not traditional holography. It's actually partially coherent or incoherent holography, which means the sources that you have in this 3D printed embodiment, you have an LED, a light-emitting diode, and it's the same in here. So you don't need temporal and spatial coherence. You just need partial temporal coherence and partial spatial coherence, and still you can push the resolution and the quality of these images to the diffraction limit, as I'll show in the next few slides. Like any other holographic technique, there has to be an engine, there has to be a reconstruction step. We're heavily using iterative techniques that are quick to converge, maybe 15, 20 iterations, extremely rapid because they're based on FFTs. This is one example. This is a very simple iterative phase retrieval routine, which tries to guess the missing phase from an intensity pattern. And once you have the complex wave with the phase, you can always time reverse fields and complex fields and go back to the object plane. This is not what we use, but this is giving you an idea of how we can do essentially phase retrieval. What we do today is a little bit more involved, and it involves essentially more constraints, more planes to be measured, which I'll show in the next few slides. With this simple design, the basic advantage is obviously field portability. You're looking at a microscope design which has just a CMOS, let's say 5 megapixel, an LED, and the sample goes from side, and it sits on the chip. From the holograms, you retrieve phase, and you can go back to the object plane over a very large field of view. The resolution of this simple design is modest. It resembles maybe a 10x objective lens, numerical aperture of 0.1 to 0.2. And the major advantage, in addition to being field portable and cost effective, is the field of view. The field of view, the area with which you can look at specimen with these kinds of microscopes, is directly limited by the active area of the sensor. In this case, there's nothing magical about 24. It's actually the active area of that 5 megapixel that we used. Most of your cell phones, smartphones, have about half a centimeter by half a centimeter active area, maybe 25, 30 millimeters square, and that's the field of view that these kinds of microscopes would enjoy. Numerical aperture here is limited, and that is mostly limited because of pixelation, because of the pixel size that we have in our CMOS or CCD imagers. To mitigate that and to create smaller effective pixel sizes in our reconstructions so that we can actually improve the numerical aperture and resolution of this platform, we've complicated the design a little bit from this initial version to something as shown here. If you look inside, it's still lens-free. It's still composed of a basic CMOS off the shelf, where the sample goes in front. It's a transmission microscope. And instead of a single LED, now we have some 20 of them. What happens here is, as the LED turns on and off to switch to another position, digitally, the shadows start to shift. And you start to sample sub-pixel shifted versions of the same static object. And the math behind this is actually called pixel super resolution, the goal being taking a small pixel and digitally dividing it into even smaller pixels. Maybe a micron by a micron can serve as 0.1 micron by 0.1 micron, and 10 of them in each direction, 100 of them total. And this is the same thing that most security cameras utilize. Actually, if you capture an image, your face will be pixelated if you're far away from the sensor. But if there are few frames that are shifted with respect to each other, then you can actually synthesize a much higher resolution image and essentially undo undersampling. This is exactly what we do. We create smaller effective pixel sizes that resolves finer features of the holograms. Some of these holographic fringes are washed away, but now they come back. And the end goal of this is essentially to retrieve, to reconstruct objects that are normally washed away with a large effective pixel size. But this is done entirely over the same field of view, which means you retain the advantage of field portability and field of view. As a result of this, you can create, even using a very old generation CMOS imager, this is a 5 megapixel imager, you can create a field portable design, maybe 150 grams total, that can have deeply sub-micron resolution and resolve features. In this case, these are thin smears of red blood cells, and these are malaria parasites infecting the blood cells, which can be reconstructed with these kinds of microscopes over very large fields of view. If you were to use a state-of-the-art imager, state-of-the-art CMOS taken from maybe some of our iPhone 4s or other smartphones, then you're looking at even more competitive results. This is actually a Sony CMOS imager. It's very small, as you can see. The green region is the active area. You're looking at about 20 millimeters square here, still quite large compared to traditional light microscopy. You can reconstruct an image that has about 1 billion useful pixels, a gigapixel, and you can reveal about 225 nanometer line widths using a pixel size that is about 1100 nanometers. That's a great advantage where you, in a unit magnification on-chip design, you can actually resolve deeply sub-pixel features, meaning that you enhance your space bandwidth product beyond the native CMOS imager dramatically, and optics is not a bottleneck there. You can even push the numerical aperture beyond the refractive index of the media that fills the space between the sample and the sensor. For that, you need synthetic aperture ideas. One way of doing this is actually, in addition to shifting the light source to create pixel super resolution, if you take the light source and have an array of them cover an arc, every time you will actually start to shift some of the frequencies, spatial frequencies that are missed by the sensor into the passband. And this synthetic aperture idea essentially takes the lower resolution passband of a vertical illumination, and every time with these oblique angles starts to cover a larger band of spatial frequencies that can be sampled and retrieved. As a result of that, you can increase your space bandwidth product and resolution significantly and essentially reach an effective numerical aperture of around 1.4 in this case. This kind of a structure also helps us with imaging of dense specimen like tissue samples. What you see here is actually a breast cancer tissue, and you can see a traditional 40x or a 20x would look at a smaller field of view, but now you have a significantly larger area being imaged with details sufficient for a pathologist to essentially make diagnosis. We did some blind testing with board-certified pathologists to show the quality of these images, and 99% accuracy with respect to a traditional light microscope was achieved using these kinds of microscopes. This is a latest generation, one of the recent generations of field portable super resolution microscopes. What is different here is that it has In addition to traditional super resolution, these are individual fiber cores, multimode fibers, that are each connected to an LED. You also have some multi-color LEDs to give you color information. And one additional complexity here is modulation of the gap between the sensor to sample so that we can actually do better phase retrieval by hopping between different heights. So you can throw in a random phase, a guess that you have. It can be zero phase or random phase. And by going back and forth between your measurements, you can actually converge to a phase uniquely, in many cases, practically, giving you essentially phase recovery in a very robust way. And if you combine this with some field corrections, you can actually create a very powerful self-correcting microscope, which also means the design, the hardware, doesn't have to be that robust in its implementation. Then you can actually do extremely robust phase retrieval. And this is shown here with another implementation, which is doing multi-height-based phase retrieval, also giving you a very good resolution. Comparison is shown here against 0.75 numerical aperture. This is the traditional microscope result, and this is essentially how lens-free amplitude images are comparing for these regions. And you have the advantage of the field of view once again highlighted here. This is the field of view of a traditional 40x objective. You can do color imaging in various different ways. Even though it's a holographic technique with single color initially, you can bring color information with various different mathematical tricks. This is a pap smear, another sample that's used for early screening of cervical cancer. One advantage of these kinds of microscopes is that in addition to their cost effectiveness, field portability, and the large field of view, they also give you 3D images. In fact, you can capture these images and send to a pathologist a few hundred miles away a movie that shows essentially how different parts of the specimen are coming in focus, which might be giving the depth of focus knob physically to the remote telepathologist in this kind of an interface. That's why these kinds of microscopes could be also useful for remotely connecting patients with pathologists because of the digital nature of all these 3D images. So far, I've been talking about CMOS imagers, which typically have half a centimeter by half a centimeter field of view. That's the typical field of view of a CMOS imager that your mobile phones have. But you can also actually work with other types of optoelectronic sensors like CCDs. In this case, you're looking at a CCD that is maybe 20 years old, but still one of the best. It's about giving you a field of view of about 18 centimeters square, where you can actually see submicron tails of human sperm in this case after super resolution. And it has about 1.5 gigapixels to it. Depending on your resolution and field of view combination, you can select different types of sensors. Depending on the pixel size, depending on the megapixel count, you will have a different combination. That's very important to highlight. The connection between resolution and field of view is broken here. You don't have the trade-off that is traditionally governing all kinds of lens-based imaging designs. If you improve resolution, you sacrifice field of view and sample volume, vice versa. It doesn't apply here. If I get better sensors with smaller pixel size and more megapixels, thanks to the Moore's law, I will have better resolution and better field of view or sample volume at the same time, which is quite unique. So far, I've been focusing on resolution and methods to improve the resolution beyond the pixelation limit, beyond undersampling limit, dictated by the CMOS hardware. But there's another element to microscopy or sensing, and that is looking at sub-micron, sub-wavelength particles or objects that do not scatter light much. For that, especially looking at sub-50 nanometer particles or sub-100 nanometer particles, we've created an interface which uses self-assembly. So what you see here is actually a trick that is borrowed from something that when we were kids we used to play. If you take two rings and dip them into a soap solution and take them apart, you will form this beautiful self-assembled shape. It's a catenoid. So what we self-assemble here using polyethylene glycol, a polymer, is the same thing but at the nanoscale. If you cut this shape in half and place a particle in the middle and shrink the dimensions to nanoscale, that's what's happening here. These self-assembled PEG molecules form a nanocatenoid lens around individual particles so that their scattering cross-section increases, which means now we can over the same large field of view of a CMOS imager, if we were to zoom in and then zoom in several hundred times, we can reconstruct sub-100 nanometer, sub-50 nanometer particles that are now confirmed with respect to an SEM image. This could be great for especially viral load measurements. Imagine you can have a device that can capture specifically and sensitively subpopulations of certain viruses, and now you can do enumeration using these kinds of field-portable microscopes. This is exactly the same thing, a little bit more enhanced sensitivity. It's a field-portable microscope. It's a super-resolution microscope. It's inverted, meaning that LEDs are here and the CMOS is there. Sample goes from the side. The sample contains nanoparticles and viruses. And you heat this PEG reservoir, which is essentially condensing and forming these nano lenses within just a few minutes. Before condensation, you cannot see the nanoparticles because it's like trying to see the moon in a sunny day in San Diego, for example, which is very difficult because of the scattering cross-section compared to the sun's direct light. The same problem, but after condensation, you start to see sub-50 nanometer particles. The smallest so far that we've seen is about 40 nanometers, confirmed again with SEM in a field-portable way. And it can also be quantitative in terms of the diameter measurement. So this is essentially not just a detector and an imager that can see sub-50 nanometer particles, but it can also count them with about 10 nanometer sizing accuracy, which is quite important because it's a field-portable design, but it can do this across maybe sub-50, 40 nanometer range, up to even hundreds of microns. So it's a continuum of particle diameters, and also density-wise, it can be a single particle per field of view, and it can be hundreds of thousands. So the dynamic range in terms of density and diameter is quite unique compared to other types of nanoparticle imaging systems. You can also do plasmonic sensing of protein binding events with this kind of a design, which in this case merges nanohole arrays for creating a plasmonic system. I'll skip this for the sake of just saving some time. So far, I've been talking about static samples and imaging them with good resolution, with good sensitivity, so that you can even see viruses. But there's another angle where computational on-chip imaging could make an impact, and that is looking at motion. Looking at motion across a very large volume. And I'll share with you some of our results on looking at sperm motion in 3D, but not just a few of them, not just a few tens of them, literally a few thousands of them in parallel, where we're tracking the sperm head with maybe 100 to 200 frames per second in 3D with submicron localization accuracy over a very large volume that is about 10 microliters. So it's a giant pool for the sperms. For doing this, we're still relying on lens-free imaging, but in a different configuration. We have two different LEDs that are on at the same time at different colors and different angles. So every time you create a hologram, you actually have two holograms per sample head, per sperm head. If you look at the raw data, this is how it looks. If you zoom in, you'll be seeing that there are lots of sperms moving around, and only if you were to reconstruct this raw data at the correct angle and wavelength combination, you can see different perspectives result from each other. And you can pinpoint to X, Y, and Z, depth information, all together with submicron localization accuracy. This is the trick to take a very simple imaging hardware and to create essentially a very powerful and high throughput 3D tracking system. Out of this, we've looked into thousands of human sperms and animal sperms. What you see here in this movie is actually a human sperm caught on video while it was conducting a helix. So what you see here is a helix. It's a very fast motion, maybe 10 rotations per second. And if you look at the front view, it's a nice circle, maybe 2-3 microns in diameter. So this is actually happening in vitro only 4 to 5% of the time. It's extremely rare, and more importantly, it moves out of focus in Z direction. That's the reason why traditional light microscopy could not see this kind of motion, because it's very rare and it's quickly out of focus. This is another one. It's a hyperactivated human sperm. It's also about a second. Now, as you can see, the circles are slightly more enlarged and irregular. These are kinds of movies that you can capture, but the throughput is the important key enabler here. We can look at thousands of them over just a few hours before the sperms die on the chip, and connect these trajectories to each other and look at everything about the motion of these microsumers. What you see here is the characterization of different parameters of these moving organisms. Among model sperms, what you see here, their linearity, their curvilinear and straight velocities of these 24,000 model sperms, only about 1,000 of them were helical, about 4 to 5%. But what's interesting here is that 90% of the helices were right-handed. Only about 10% of these helices were preferring left-handed motion. This is something that we do not know why, but it's quite interesting and funny that it coincides with the percentage of left-handed people. About 10% of you, according to Wikipedia, should be left-handed. And there's no correlation, obviously, between the left-handedness that we talk here and your own left-handedness, but it's essentially an interesting observation. Since then, we've discovered other types of interesting motion. This is actually a horse sperm conducting a ribbon, like a sinusoidal motion over a plane, but the plane itself is twisting. This one is going to be a movie of a horse sperm conducting a helical ribbon over 4 seconds. Just for the sake of time, I'll fast forward it and show you essentially its motion. It's a plane dictated by a sinusoidal motion, but the plane itself is beautifully curving like a ribbon does. This is the first time that any type of microsumer has been observed to show this kind of motion. This is another one. This is twisted ribbon. It's also beautiful. These are highlighting some of the powerful features of on-chip imaging for not only microscopy, but for also tracking motion. This one is also a great mathematical function. This is actually a helicoid. It's a minimal phase function. It exhibits zero net curvature, just like a catenoid does. So in that regard, there's some interesting math behind it that we do not know why sometimes they switch to these kinds of beautiful mathematical functions. I want to come back to this slide where I started. All of this is enabled by the rapid growth of our CMOS technologies that is powered by economies of scale driven by mostly consumer devices and in particular mobile phones. Thanks to us buying more and more mobile phones more and more frequently, we've now reached more than 40 megapixels, and this enabled us to improve our space bandwidth products every 2 years in double the number of pixels that we can actually create. And using some of those 40 megapixel imagers, sensors, and smartphones, we've actually also pushed fluorescence imaging using a mobile phone and created dark field and fluorescence microscopes attached to mobile phones as shown here in a field portable design to look at individual viruses. This is actually a design that can look at glycoprotein labeled individual viruses. What you see here on the cell phone image is actually human cytomegaloviruses, about 100 to 200 nanometers in width compared to a confocal microscope image of the same. Also could be used for specific and sensitive viral load measurements. And recently we pushed this to looking at DNA, single stretched double-stranded DNA molecules. Actually, this is a cell phone image which shows you individual DNA molecules that are stretched with disposable microfluidic devices, and then the image is shared with our servers for measurement of their length. Our length measurement accuracy is about 1 kilobase pair for 10 kilobase and longer DNA strands. Then you can start to do genetic testing, and you can look at, for example, copy number variations in the backbone of the gene, which could be quite applicable for especially looking at drug resistance in malaria and TB, something that we're working funded by NSF. This is another work that I very briefly mentioned in the introduction. This is a fluorescence microscope coupled to servers for machine learning. It's not just capturing good quality microscopic images of Giardia particles, waterborne Giardia particle cysts, but also capturing these images where Giardia particles look like these rugby balls elongated, and then at the server backhand we have a machine learning code which essentially counts them and reports back to the smart application on the phone. A similar interface has been used for clinical testing. These are ELISA tests that are read in microplates. We created recently a mobile phone attachment for 96 well plate reading. So there are 96 different wells here which are through fiber optic cables sensed in transmission and imaged by the cell phone camera. It's a very large plate. As you can see, it's a centimeter scale bar here, and this contains 96 patient samples to be imaged at the back of a fiber optic cable array, which then is also sent to our servers for also machine learning for quantification of the diagnostic test. We took this actually to a UCLA clinical microbiology lab and tested this with about 1,100 patient data and achieved about 99% accuracy in all these tests: mumps, measles, HSV, human simplex virus 1 and 2. These 4 tests are clinically approved, FDA-approved tests that are being used for immunity screening. These kinds of interfaces could be quite important, especially if you want to screen, for example, immunization and vaccination campaigns. To sum up, I've talked to you about some of the work that utilize computation for simplification of traditional designs of microscopes, sensors, and diagnostic tools, some of which are used in our lab settings for treating patients. And I've shown you and given you examples of how some of these designs are catching up with the performance, with the sensitivity, with the specificity observed in clinical settings. And in some cases, like in the microscopic imaging examples that I've given, it's actually enhancing the designs of traditional instruments, measurement devices. And that's where I think the democratization angle comes into play, because some of these devices would open up new opportunities for educators, for engineers, for medical personnel in resource-poor countries. With this, I'd like to acknowledge our funding agencies and all the graduate students and postdocs and undergraduate workers in our lab behind this work. Thank you very much.